Today, I'm going to give you an introduction to the idea of protein function, and that will be about personalized health. It's particularly convenient for this subset because for you it will be completely new. Oh, I don't know why. So we by now know that Jonas is going to do the recording, and this is the next week. Tobias Hamp is going to talk about protein function prediction. This week, again, intro into the lecture. Okay, by the way, since you're all living in Munich, where is that? All four of you. Any idea? So it actually sort of is a trick story because it no longer exists. There's the English Park, English Garden, and there is this house at the lake where you in fact can eat. So this is like a beer garden. And there was behind that there was the public viewing of football in summer. Weren't you there? No. But this figure.
I would have said English Garden, but
this figure has disappeared. So the title of this talk is missing, which is beaming from SNPs to personalized health. And beaming, you all know the idea of beaming. You know the Starship Enterprise? Okay, so then you're familiar with this idea of beaming. Beaming is for me something that, whenever I'm caught at some airport in the world, I dream of. So one day this is going to happen. And when I'm in a bad mood, I say, I suspect that this day will be after I retire from traveling around in the world. So can we do the same thing? So can we have a fast forward track from identifying SNPs to really personalized, individualized medicine or personalized health? Let's get to underneath what the idea really is. You want to map what people call the genotype to the phenotype. Any idea what these words mean? Let's begin there. Give me an idea what this word means. Fair enough. Yeah, you're right in the right absolutely in the right direction. It does not have to do with an old typewriter. It has to do something with the gene. Any idea what more? Anybody else?
instead phenotype could be.
Let's begin here. Something with the genes, yes. So the idea really is the particular aspect that is in your genome. The genotype, essentially one idea that we have in our head is this is in fact your genome, individual genome. And genotyping is identifying your genome. Okay, let's get to the other word, phenotype.
Could it be from the phenomenon? Maybe words, so
as an Italian who knows Greek. Thanks, John.
Maybe it could be something related to the interaction with others.
That's a little bit of a far interpretation. So your Greek was not that firm then.
It's something that appears.
Yes, but there's not necessarily an interaction. Exactly. And that, in fact, phenotype is the way you are. Okay? It's one of phenotype in biology has all kinds of different meanings. Ultimately, it means something in the original Greek definition, something that appears, something that you can measure. Something that leaves an imprint on nature that makes a difference. Okay? And phenotype can be all kinds of aspects. For instance, I have well, in this particular case, it's sort of grayish hair. But at some point I had blonde hair, or that would be a phenotype, okay? So the way I define myself in the mirror in the morning, there are an overlay of many phenotypes. But the question ultimately is how one particular aspect of me, or how the whole thing relates to the genome, right? And in some sense, that's what these images here mean. That, in fact, is the genome material, and we can sort of gaze at the genome material at your hand, and we learn something about you. To treatment. So going from, well, some people can read hands, right? And it's somewhere written there. with or without the modern biology of genotype. So the idea here that is illustrated in this drawing simply, and I'm sorry I don't have a source for this drawing, is essentially we have different people and we have different drugs. And somehow the background idea is we map one upon the other. And I will revisit that statement. Personalized medicine would then somehow be something that has to do with, it begins with your genome on a chip. Somehow, right? Your genome is there, that's your genotype. And then something, when we read that, something will happen in response to that genome. That something could be a particular treatment. And in order to get to that point, we clearly need bioinformatics in order to, in fact, understand the data underneath. And we need a ton of different methods shown here in this server predict protein that, in principle, takes in a sequence and predicts all kinds of aspects of protein function and structure, and that, to some extent, is what we're going to go through in this semester. And we want to have it on the level of DNA, RNA, the biomolecules, on the level of protein signaling, cells, organisms, the environment. We want the whole exchange. And that's where the interaction gets into the play. And that's why, in principle, I removed the Greek word phenomenon from the interaction. Because very quickly, the story gets much more complicated. But the phenotype is not necessarily defined as this interaction. It's part of it, depending on what your question is. Okay? So let's begin with the amount of sequencing that we know. Sequencing complete organisms. So let's get back again for those of you who haven't had a big background in the field. So there's DNA, information code library, RNA, intermediate step, and then there are proteins. That's they are the machinery of life. DNA comes in this legendary double helix. Proteins come in different shapes, and these shapes we will get back to in a minute. Now, does anybody, oops, I'm sorry. You all can see it and it's too late. So, if I had asked you, would you have known what the first entirely sequenced organism was? Yeah, now you're fair enough. They all look up. Come on, I made a mistake. Give me a slack, give me a break here. The first one was in fact Haemophilus influenza. Was published 19 years ago. And that was a publication, a tiny bacterium, shown here. It's a micrometer about. And it was published by 40 authors. The next one, in fact, Mycoplasma genitalium, is bigger, is smaller, sorry, it's a very, very minimal organism. And the first eukaryote was yeast, baker's yeast here, a year later, as you see. The first sort of animal is a worm, C. elegans, and then comes the fly, Drosophila, in 2000. So within 5 years, you have essentially a bunch of organisms sequenced. Now, what about sizes? So Mycoplasma has about 500 genes. Haemophilus has about 1,700, so that's a bacterium. Methanococcus, and there are all kinds of mistakes on this slide. Methanococcus is an archaeobacterium, an older bacterium that survives in extreme circumstances. Actually, in terms of numbers here, is very similar to the bacterium, but you already see that there's a spread. About 4,300 here for another bacterium, E. coli. And then we move on to eukaryotes. Yeast is a eukaryote, but in fact it's single cell. It just lives as single cells. That is the next step here, for instance, to Drosophila with about 14,000, C. elegans 19,000, Arabidopsis thaliana is a plant, 26,000, rice 50,000. And what people believed in 99 was that human has 100,000 genes. Now you all know that the human sequencing was done. It took about a little bit over 10 years and was supposed to be completed in 2000. And then there was this meeting. In that meeting, at the end of 99, it was revealed that it would not be on January 1st, 2000. And it was revealed that this idea of 100,000 proteins is not precisely right. So again, they wanted to have the genome published on January 1st, 2000. On October, 3 months before, after over 10 years of project, they say, okay, it's not 100. We made a tiny mistake. It's 30. Right? That's a tiny mistake in biology.
It's a tiny mistake.
For biology, that's a tiny mistake. That's the point. That's you're in a field where 3 months, after 10 years of the project, 3 months before, the belief is 100,000. In October, it's suddenly called a tiny mistake is only 30. Those are tiny mistakes in biology. That gives you an idea about the size of the steps. What happens in biology is amazing. Now, how come that all of this came so shortly before the genome sequencing? The fact is they had to assemble all the data. And that took them over 6 months of CPU time. And before that, they had collected all these data. But they still assumed that this number would be right before they did the first reassembly. When they had the first assembly, they questioned that number, but it couldn't be. Their computer must be wrong. Sure, everybody believes it's 100,000, and you compute that it's a 30,000. Nobody's going to believe you, so you're going to do it again. That was, in fact, one of the major reasons why it was delayed, because what they found couldn't be true, right? And you saw here, then that's the public announcement here with then President Bill Clinton, Francis Collins, and Craig Venter from the private company. And then the idea was that essentially there was a public project from the NIH and other funding agencies. Sanger in the UK, in England, was very big on it. And a private company, Celera, by Craig Venter. And both of those sort of came at the same time, roughly. They sequenced a group of 5 people. The company sequenced 1 person. And it's your guess who that person would be. Okay, so those are the slightly changed genome sizes. In fact, today we believe that the number of human proteins, genes, is less than 25,000. So the latest number is more in the ballpark of 20,000. Any surprise here? Right? This is twice as big as a human, right? I mean, rice, so for all of you who like to eat rice, there's a lot in it, right? Much more than in us. That means something, right? So how come that we get this darn little C. elegans worm, which has 900 neurons, right? And it has a similar, almost a similar number of proteins as we. How can that be? How can it be that a plant, well, a plant is slightly different. Plants can't move, so they need some additional material for protection. We can sort of understand that. But still, in principle here, between C. elegans and human, that's somehow similar evolutionary conditions. But we still are much better, much bigger, much more important, right? How can we explain that? And the answer simply is we still don't know. That was the other shock when this was published. The early idea really, and I said that for the Celera example, was that we essentially need one genome, and whatever we see here in this room would be a tiny deviation from that reference genome. So we take any of the people in the room here, and that could as well be the director of the institute in Celera, and then essentially we would define all of us as tiny differentials. That was the idea in 2000. We have 23 chromosomes, so that's simply the way the genome is organized in 23 chromosomes, and they are 3.4 gigabase pairs. The genome size, again, this is similar image here for the number of proteins, but this really is the base pair, the raw size. When I told you proteins, so in some sense those are the units. Does anybody in the room, I'm not entirely sure, I'll get back to that, in the room know how much of these 3.4 gigabyte in human is actually coding and leading to proteins? So I said the proteins are the machinery of life. What fraction of these 3.4 gig leads to a protein? Any rough idea? Half of it? Who's happy with half? Who would say less? Who would say more? Less. Anybody wants more? Okay, let's follow up on the less lead. Who would be happy if I said 1/10? You would be happy. Anybody not happy? Who has a general outlook to life to never say anything? Is that all the rest? Okay, so we currently believe that it's in a ballpark of less than 1%, or in the ballpark of 1%. So most of that, and that's this word out there that you may see in the press, that is why it was coined, most of our genome is junk. We absolutely do not know whether that is true. And in fact, there's a lot of evidence that this is absolutely not true. But what we are working on in this lecture is essentially about 1% of the genome material, right? And that is so far, for what biology knows, the most important part. All your drugs are targeting exactly that fraction. One way of explaining how important that little percent is. Okay, now let me skip that one. And I'm not sure why, sorry for the animation. Now we have the parts. We know the genome. Do we understand how the organism works, right? Do we understand the whole thing? And in some sense, it is, Anna Tramontano from La Sapienza says, like for every good manual, just imagine the genome is the manual, right? And like for every good manual, you have the manual, but if you have the manual, you hardly ever find what you're looking for. And let's face it, at least for me, whenever I find it, I don't understand it. This is a good manual, works like that, right? We all used to that. Somehow with modern programming, the idea is that the programs, you don't need a manual anymore, they work by themselves. So that is a big step. And to some extent, you seem to be laughing, not believing it, but I come from a different time. So 10, 20 years ago, reading software manuals, it was horrible. And just the installation instructions, some of you may still use these things, what are they called? Windows or something like that, where every single time you install a program, you have to go through 10 pages or something. Incredible. I'm overstating the case. Anyway, still, the fact is true. Imagine you took this thing apart here, and then somebody gave you all the parts, but they wouldn't explain to you what the parts do. And now your task would be to put it all back together. And in many ways, this is the situation we have. Because we cannot even label the parts. And we get back into that. And it's not only this little car here. It is much bigger. Can we compute life? It's a tough challenge. And we address it from all possible angles. But we still really don't understand the book. Although we don't understand the book. And that back to Craig Venter over 10 years later. You can have life in a test tube. So you can create an artificial organism. That essentially you take, what I showed you is a very tiny organism, Mycoplasma genitalium with 500 genes. You look at what of these 500 is a small number. You can look at them one by one and say, well, those may be special to Mycoplasma, those may not be important, strip it down to something that is much smaller than that, and then take that as the base to build your own organism. That's exactly what they did. We still don't know what these proteins all do. We still don't know how life works, but we can produce it in a test tube. And again, as a result from Craig Venter. And I believe that computing life is an important dream. And I believe it will help us to get closer to it. And this is in some sense what we're working on. Let's look at the issue of mapping the genome diversity. I said one genome was enough. And by the way, 3 billion, 3.4 gigabases. What it means is 4,000 books, okay? This image shows exactly 4,000 books. I counted them. You may be laughing, it's really true. I took books out of the shelf. Just, every letter in here is one base, that's your genome. It's an amazing number, just picture this. And underneath, again, are the proteins, are the machinery of life. So the HapMap project and then the 1000 Genome project were projects launched in 2008 that tried to understand the diversity. Okay, the idea still was somehow there is a reference genome, but let's just see how much diversity there really is out there. So let's sequence 1000 people. Okay, that's 1 kg. And I'll get back to the data. Later came 10K UK, and in fact the Chinese now sequence 1 million people. The BGI is the Beijing Genome Institute in Shenyang, and that is by far the largest sequencing center in the world at the moment. Okay, so what can we, how much do we differ? Does anybody in the room have any idea?
20,000. 20,000.
You're too fast. You have these eyes that yeah, come. Well.
Is the class always here?
Yes.
Tom online was confusing us.
Oh, I'm sorry. Yes, and we changed the room because the group was too small. And then we felt, various people felt that, thanks to, hi, come in.
Oh, no, it's for you.
Come in, you're right.
He's a friend of mine last time also. We went there 2 times, but we were not sure.
That is, however, the last 2 times is my mistake because I was sick. So, but the lecture will always be in this room.
Great.
Thursday and Tuesday. Okay, so she saw the answer, 20,000. 20,000. Let's, oops, the wrong button. Is 20,000 a lot? Let's just picture 20,000. 20,000, that's the Bavarian coat of arms. We differ by 20,000 amino acids. So amino acids are the building blocks for these proteins. Okay? And we have 20,000 proteins roughly. So in every single of our proteins, every pair of us in the room here has 1 amino acid difference. That is sort of indicated here by what we have in common is blue, and what we have in what is a difference is red. Okay, now look at this coat of arms. Do you see a lot of red here? But you forgot one thing. Every single cell in me has exactly the same 4,000 books. Every single letter in every one of these 4,000 books is the same. In all my cells. Well, I'm aging and things should change a little bit. But other than that, in principle, that is true. Every single time a cell duplicates, cell splits in your body, and it happens during the day, constantly, you get the same 3.4 gigabases. No change. So behind that context, this is huge. This is much, 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 much more than was expected. And in fact, in some sense today, in terms of sequencing, biology has become the age of diversity and variation. And we can learn from it. So here is a totally different part. Tick. This is the best feat I've ever seen. This is remarkable. How did you do that without topping it over? Amazing. So I talked about the genome so far. Epigenetics is something that comes on top of the genome. The genome is what you inherit. The genome is what you carry all your life. So when cells duplicate, there are some mistakes. Radiation brings in some mistakes. All kinds of influence from the environment could influence some mistakes. But in principle, your genome is the same. We all come from a single pair of cells. Those, however, are not heritable changes. Epigenetics are markers on top that happen during your lifetime that in fact may influence who you are, how you feel today, on a different level, on the super genome level. There clearly are important influences, and we will not talk about that in our lecture. I just wanted to mention it. One way is in fact the way the DNA is shaped. This influences the way the proteins are expressed. you could imagine that if the kink is a little bit too much or too high, then that protein could not be accessed. Or there would be some reading mechanism that couldn't see that protein. And that could be a change that in fact happens through your lifetime that you pass on directly. Okay, so from this diversity we can discover all kinds of issues. One, for instance, here was that there was a migration to North America before the contemporary Indians, or before what we actually believed was the first time that people went to the Americas, which for those of you who still believe that is Columbus, is a long time before Columbus. But still, this is much longer than anybody had thought. And the way that was found out is through sequencing. A piece of a body found in the cold. Here's another story, and that comes from Svante Paavo. There's a cave in Siberia, the Denisovan cave, and in this cave they found a remain of some animal that turned out to actually be a human, and turned out to be a new species of human defined through sequencing. It's the first time that a species has been defined, in pre-human species, the Denisovans, have been defined through sequencing. Human diversity. So now let's begin a couple of these simple things. Let's begin here, because there's a party interested in this sort of picture here. You know, they are all Italians. They are all great football players, and you see that there are differences between them. They have different height. They have slightly different hair color, and the same is true here for the Swedish team. But wouldn't you all believe that if I asked you, if I look at any pair, oh, I lost my pointer. At any pair, oh, because it's empty. Any pair of people here, and compared their genome with respect to anyone here, wouldn't you believe that they, despite all their differences, are more similar to each other than any of them to any of those? They're all football players. Or do you believe this is sort of making them more equal in terms of their genome?
Human beings in the end.
Yes, that is not my question. Oh, I'm not expecting a fish or something like that. But the point really is, if you met them on the street, this group and that group, and you passed on, you could immediately say that was the Italian team and that was the Swedish team. And most likely you could do it for every single person. Maybe you make some mistakes, but in principle you could do that, right? Don't you all believe that?
Yeah.
And that should be visible in their genome, right? Sure, they're all human. Okay, if you're not convinced, so how would you then say, okay, they differ. They're all European. You said they're human, but they're all European. What about this team? So with respect to that you keep, don't you believe that they are more similar to each other? Now everybody's happy. So let's get to the next one. These are Bushmen in South Africa. And they all are very much shorter than I, and they come from a certain region in Africa. And again, when you see them, you can clearly say, those are the Bushmen, and they're not from the Chinese team. You will never make a mistake on this, okay? Now, you would clearly assume that despite the differences between the European and the Chinese, they may still be more similar. These guys here are much more similar to each other than any of those to these. And that all is wrong. Every assumption here. You're the only one who doubted the first one with a I don't know why. The Swedish obviously buy a lot of Italian players or whatever the background here is. But everything I said was wrong. The highest diversity is here. These guys, these Bushmen, differ much, much more from each other than I differ from a Chinese, or than I differ from anybody in the room here. Not sure about your background, but...
You mean at the gene level?
Yes.
Okay. Not in looks, hair type, or whatever?
At the gene level.
Oh.
That is correct. But that's exactly where looks in that sense, are deceiving. When I look at the genotype, when I look at the genome information, what we see is not what is imprinted in our cells, in some sense. And ultimately, biology is a lot about post-diction in some sense, about you observe something and then you pretend that you always knew what you would observe, why you understand it. In this particular case, the way to rationalize that observation, that was a big surprise when it was published, and I don't have the paper on this slide anymore. Well, it's not it's not really readable, but is that paper here? When it came out, that was a surprise. The way it's rationalized is most people assume, most scientists assume, certainly most scientists in the Western world assume, that mankind came out of Africa. The beginning of mankind is close to where the Bushmen are living, to South Africa. Now, imagine you have a population. In that population you have a certain diversity. Now you take a subpopulation. Now let's ask the first question. You have the population, the diversity in the population, and you take out a subpopulation. Which of these two groups has a higher diversity? Has a higher diversity? No, how can that be higher?
It's lower.
It's a subset. A subset will always have a lower diversity. Because in the big set you have all the variety you can possibly find. And then you take any group out at random. No matter what, you will always have less than all, right? I mean, I'm sort of trying to push the point. But the diversity cannot be higher in the subgroup. It can only be lower. So that is the rationalization now. We take a subgroup, go with that subgroup out of Africa, and then split again and split again. And every single time somehow the diversity is reduced. That's the idea. But again, with this being the idea, still the result was surprising. So let's go from gene variation to protein variation. SNP, and that for those of you who are new to my lecture, you really do not, I want you to understand the idea of a gene. I want you to understand proteins. It is very, very helpful if you knew the word amino acid. But most of these words are not absolutely needed, okay? And this is in particular one that is not absolutely needed. Still, the idea behind is important. SNP originally stands for single nucleotide polymorphism, and the idea really is, so those are is the double helix. The DNA has in fact 4 different letters, C, G, T, A, and you see that a single letter differs. And this single letter difference is called a SNP. But historically, this was only called when there was a certain number of changes in a population, because our sequencing techniques were not good enough. So we always said, if a particular letter changes in at least 1% of the population, then I call it SNP. Else, I call it a sequencing mistake. That simply is the origin for that. And since the sequencing technology became better, this is no longer really the point. Still, people talk about SNPs. Non-synonymous SNPs are those that change the amino acid. So again, we have the DNA, 4 nucleotides, translated to 20 amino acids, and they form the proteins. And things that in fact change the protein sequence are so-called non-synonymous changes. Okay? When we look in diseases, here we have non-synonymous, the ones that change the amino acid in green. Red, synonymous, those that do not change, for the different human chromosomes, and this is a raw number. And you see that in fact for the proteins, so again this is the 1.5% of the entire genome perspective, the 20,000 proteins, all the things related to disease, in some sense known through a particular type of study, are shown here. Most of those are in green. Most of those in fact change the amino acid sequence. And some of those do not change the amino acid sequence. So I said proteins are the machinery of life. How can you imagine that the red ones that do not change the amino acid sequence, that in fact create the same protein, how would they lead to a disease? And the answer to that is that has some regulatory and unknown explanations. Mostly that is there. So sometimes we can see that this, in fact, before the protein becomes a protein, this change leads to the fact that the protein never becomes a protein. That could be one reason. Why, for things, the moment they become a protein, they should still matter, it remains unclear at this very point. Here is an image of different amino acids, but in principle, personalized health or personalized medicine means that we go from this genotype, from these changes, to different drugs. That's the idea. Now, how far are we on this idea? Let's begin again. There are different issues. There are epigenetics that I suppress here, but let's assume that would not be important. GWAS, genome-wide association studies. So it's one way of, in fact, looking at the differences. So you look at the population that has a certain disease and a population that doesn't have the disease, and from that you essentially try to associate which ones are the SNPs that are related to that disease. Okay? So we do this. We do that for many patients, for many diseases, for many drugs. We take into account environmental factors, viruses, bacteria. We map all of this to the interaction of genes, proteins, drugs, environment, put that into a simple pipeline that is readily available for clinicians, and the job is done. In terms of sequencing, we can easily do that now. Again, the BGI is now sequencing 1 million people. So we could in principle do that, yes?
Might be a foolish question, but in this term, would we read that there could be better genes which are susceptible to something, not to something?
Yes, absolutely. So we can, I will give you later an example for such a case where a particular mutation has aspects that you would call better. It does something and it does not do something else or something like that. Yes, but let me go to the end of the lecture. No. Let me make this point again. There's absolutely no foolish question. There's the idea that a question can be foolish is completely misconceived. You're open to ask any question. But this particular one I suppress. I'm not answering because in some sense ask it at the end if you still feel like it. So how much of that have we done? Let's begin. There are many challenges. The top-down challenges is the killing power, the killing of numbers, and this is outdated here. There is an Illumina High-seq that is no longer contemporary. I don't even know when I made this slide, but it's 3 years old for biology. That is no longer valid. 3 years is a different century in biology. Still, that machine, different century, right? Produced on essentially per day one human genome, and it produced at full capacity 25 terabyte every day, one machine. It's a big robot though. Now here is something, or let's get to the next level here, the MySeq, and again it's outdated, it's much more than that. This machine here looks exactly like a desktop printer. It comes in the same package, costs a little bit more than a desktop printer, it's 200,000. That does, in a full day, 10 terabyte. Okay? A desktop printer that produces every day 10 terabyte of data, just imagine this. So how are you going to get that? Now each one of us is going to be sequenced here in a day, and now you want to ship that to your collaborators at the Broad Institute. How are you going to do that?
I don't know whether these machines will be effective for these types of data processing.
The machines produce the data. They are not effective for the data. But now you have you read it out of the machine. How do you get it to the guy in Boston?
The foolish thing will be to transport it.
How?
Like just with any means of transportation?
Yeah, what? No. Tell me what you believe. How will it be transported to Boston? We are 2015 or 14. So modern internet. So how are we going to ship this to Boston? That's my question. You get back to yours in a moment, but please answer me that. How do I get that to Boston?
Cloud database.
I mean, the
10 terabytes? How do you get 10 terabytes through the internet? And you do that every single day. You know what the LRZ is going to do to me if I pump 10 terabytes every day in there, or try to. I'll never get it through.
It's better maybe.
Even if I had the standing line, that is not easy. So the current procedure is?
The grid maybe?
The grid. No, how do I get it through the internet? 10 terabytes.
Chunks?
Yeah, chunks. Still, after 24 hours, 10 terabytes. And I do that 365 days. No way anybody's going to be happy with that. A petabyte? So how do you believe the data is transported? And that's a tiny machine. It's a desktop printer, goddamn.
Printed them.
4,000 books. Okay, printed. You go into shipping. I mean, you need it. No, but you're close to it. They send it by FedEx. Yes, they actually ship the disk. In 2014, what did they end up doing is shipping by FedEx disks. That's the fastest way to get the data out. Anyway, so you had a different concern. What was your concern?
Maybe that was like to use a grid system which can work.
No. So that is in fact what I'm trying to illustrate here. These numbers are really big. And that's a tiny machine. So the ones that I showed you before, in fact, again, it's outdated. The Genome Institute in Shenzhen had 147 of these machines, and they run full time. There's not a moment that they are stopping. Just get an idea of what these guys produce there. That is big data. And it's big data not in the future, it's there. And every day more. Now, here's another issue that is a big problem, and that is sort of understanding in the top down. So biology is organized in different groups of people. So there are some people who are experts in proteins, and some people are experts in genome. If I want to go from these diseases to the proteins underneath the machinery of life, all of these people have to communicate. And I can tell you, this is really complicated, even with friends, because we speak such a different language. Then there are these big unknowns. We had this before. What is a big step in biology? So the idea really is that's only the information. There's an intermediate step, and then there's a protein. We now know there's a whole universe of things in between here that have an active role in the process, in the way life works, RNA. And we still really don't have unraveled how that works. Then there's this issue of the bottom-up. Bottom-up means annotation, for instance. So when I get back to the level of the proteins, this lecture is about predicting protein function. How many proteins? So we have 20,000 proteins. For how many of those do you believe we know function?
Most of them.
That's absolutely what everybody else would expect. I'm telling you, the answer is for 10 to 50%. Okay, 10 to 50% in your book may be quite a range, right? So is it 10, is it 50, or why can I tell? And in some sense, the question is, if I call this here all red, then it's 50%. If I call this all red, then it's 10%. So meaning the level of coverage that I have. And in a similar sort of analogy in some sense, if you call that an aeroplane, then we have 10%. If you call that a clear image of an aeroplane, oh come on, that's a beautiful plane. I mean,
I thought it was a smile.
Yeah.
That's the artist. That is a plane, isn't it? So if you call that a plane, and you're happy with calling this as much a plane as this one, then we have 50%. So that has to do, essentially, the range has to do is how much detail do you want, if I ask you the question, what do you call I know function, right? And if really this level of detail is what you want, then it's more in the 10% category. And this, in fact, is what our group does. It tries to predict protein function from sequence. So let's get back to this issue. Genotype to phenotype. My genome, or the differences between our genome, to phenotype. for instance, to the type of diseases I have. Personalized medicine, one connotation is there is a drug per person. Is that realistic? So you essentially, you take your genome and go to the doctor and get your drug for it. Is that realistic?
Only one drug.
Only one drug. Is that a question or was that a statement?
Yeah, no, I'm asking. I mean, it makes sense.
So the idea that you have a specific drug for your genome, for yourself, is that a realistic idea?
I don't think so.
Maybe.
Why not?
Because it's a lot of resources for each person, and we cannot afford it for now.
Do you have any idea, does anybody in the room have any idea how long does it take to develop a drug? How many drugs are put on the market?
2-3 years.
Yes. Maybe take years to develop a drug.
Years. It will take years to develop a drug.
After it's developed, maybe just mass production.
We will get back to that. I will give you some more detail and we'll show you that slide in more detail. So today it really is a run through some of the themes that we will cover in more detail later. Don't try to understand every single part. It's like I give you a flair for it. On average, it takes 13 years. About 13 years to develop a drug. What does it cost? Anybody? Ballpark number?
Lots of fruits.
10 million?
You're under. 2 billion. And so Marco De Vivo, there's a talk from Marco De Vivo that we may see live. I don't know whether he is going to come. This is the number of new drugs that are added per year. And you see it's in the ballpark of 40 over the last 15 years. So the idea, one drug per person, is absolutely not realistic, right? This cannot be. But we can still map the genome, the genotype, the way my genome, the differences between us, to a particular disease for diagnosis. Or we could possibly use it, in fact, to choose the drug. That is feasible. I'll get back to that later. In order to do that, however, we would have, again, every pair of us has about 20,000 amino acid differences. So we should first find a way to distinguish between those that matter from those that don't matter. For that, we would have to predict the effects of changes. And some of you have seen this before here, and I'm going to show you in 10 days again. Oh, actually, I'm going to get part of this on Thursday. This is number of residues aligned between two proteins. This is the number of amino acids they have identity. If we have two proteins, each 100 residues aligned, so we have two proteins that over 100 residues have 33 or something like that in common. Then, this is blue, they essentially have the similar three-dimensional structure. Okay? Can turn that around. 33, 100 minus 33, so you can change 67 and still maintain structure. You maintain some aspects of the protein, so not every single change most likely is going to lead to an effect. How can we distinguish one from the other? And how can we in particular predict the effect of a single change? And that has been done by Jana Bromberg. When she started her PhD, they essentially, and again, I'm going to go through this in more detail, there were 3 different methods, and they all essentially looked at the mutations that I see. Have I seen them in other organisms? If so, I say they don't matter because other organisms live with that. If I haven't seen them before, maybe that matters. And she wanted to do the next step by putting machine learning onto it. Machine learning means you have to use a lot of data. A lot of data means that you in fact have to average over different sorts of data. Over data where you say there is a mutation that makes this difference. And in the other extreme here, where you have a binding reaction between two molecules, and you observe a different binding constant, and you say that now is a difference. And you immediately see that here, these two differ is immediately clear, right? They have different colors, or one has fur, right? One is naked. While here, binding energies, you all have had enough chemistry to know there always is a spectrum. So you have to define a threshold to say binding, not binding. And there is no, in chemistry, there is no on and off really. And most reactions at least have a steady state in between that is somehow populated. So there's different sort of data. And then she gets a method, and that method works to some extent. And I'm going to just skip over these parts here. But let's now go back. So the method can predict the effect of a single amino acid change. And it can do that to some extent well. So now let's look at Neanderthal, chimp, and human. So the same group that I mentioned before, the group of Svante Pabo in Leipzig, and the data comes here from Janet Kelso, they sequenced the first Neanderthal. Any idea what the problem is of sequencing Neanderthal? Exactly, they don't run around that much. It's not a very frequent sight at the airport. By the way, I want to point this out, the artist rendering here, so the way we picture these Neanderthal has changed over the time. So where do you get it? You have heard of the Neanderthal, I assume?
Glaciers.
In
the glaciers.
Yeah, well, that was not a Neanderthal. You're talking about the Italian. That was 5,000. You talk about the guy who's in Bologna now? Bologna. In Bolzano now? Or what? Ötzi?
Maybe I will.
That is a different time. It's a very, very different time.
They are with bigger brain than us. They were different.
They were a different species.
Yeah.
Okay? True. The brain, and again, so my point here, artist rendering, we don't quite know how they looked. We haven't met them. But again, how do I get their genome, is the question here. So Svante set out, in fact, to compare hominids and apes, and simply from studying the sequencing, learn about what makes us us. And one simple point to start is a different human species, Neanderthal. We are Homo sapiens, they are not Homo sapiens. They're Homo neanderthalensis. So, but how do we get the sequencing of those?
From the fossil?
Exactly, that's what everybody would assume. Can anybody sort of suspect what the problem with that is?
We've not found a fossil.
That's not the problem. Actually, there are lots of, almost every good natural history museum in the world has some bones of a Neanderthal. And some of them have almost entire skeletons. So Neanderthal is still is something where you have many, not like thousands, but many. Okay. So just drill into those bones and get the sequences, right? But it doesn't work. Or it ultimately is what they did. And they published a paper, it's not shown here, and the data that I'm going to present here it comes out from that paper, from them. So ultimately, in some sense, they did it. But what is the challenge?
They're not living.
That is certainly a challenge, yes, again. And that would also help for making this photo here, if they would be living. No, but, so I have this bone thingy there, right? I have a skeleton. From the skeleton, so I assume they found a guy who's 5,000 years old and was frozen in a glacier in the Alps. So I take that skeleton, take out the DNA, no problem whatsoever. Here, the story is more complicated. Why?
Maybe you can have to get DNA from the fossils.
Why? What's the problem?
Because the tissue is completely absent at him.
So you get a lot of DNA, just not the one from the Neanderthal. So you get a lot of bacteria, that actually a lot of the other animals that died over this site. And
So they mix
you get a lot of human. Why did you get a lot of human? And that turned out to be the major problem of the one of the major problems.
You get human DNA from the.
Yeah. Why? because they wash their hands. So the humans, in fact, handle the bones. So, and that's the most active DNA that you extract from the bones of the guys. And the more clean, so the newer ones were in fact the ones who were more polluted because they washed their hands more often. So more of their stuff got in there. And in fact, the finally successful bone was one that he found in a cave, not in a cave, in an old museum in Croatia, because it had not been touched for 100 years. Just no human had handled it, and somehow that was the cleanest. The next issue really is that genomic material is falling apart. The genome is falling apart, and that has to do with enzymes that are in the air. The air is full of living material. It is the flu, and it is the viruses, it is what made me sick last week. But that does all kinds of things, and it also eats up the genome material that you put down. So that's another challenge. Now, one data set that they looked at is they asked, if I look at all the sites where Neanderthal is similar to chimp, so the amino acids, where Neanderthal and chimp have the same amino acid, and where both have a different amino acid than human. Okay? How many were those? When we started this project here, and Rost started this, and Yannick Malik, we had actually 78 amino acid changing mutations. So again, between every pair of us, we have 20,000. That was a set of 78, a tiny set. And with this tiny set, we tried to, in fact, see what we can learn from these 78. We didn't get very far, but we're still working on it. Let's look at the population of the 20,000. Can we say that the 20,000 that we change, that are between us, have an effect or not? If we did the experiment, maybe in 2050 we could answer that. In the meantime, we really need the computers, and that is the method from Jana. That method, again, it predicts, it's a neural network based method, so you have a variant as an input, and then you predict whether or not, to output units, there is an effect, or it's neutral. So assume that this is sort of the thing you come in with, and now shape stands for function here, just visually. So that is the same thing. It does the same thing. So that's a neutral change here, while this may have no function, and this may have a different function. So effect could be two different things. Okay? So now what I'm going to show you is cumulative plots, where here in that direction, the method predicts a very high effect. In that direction, it predicts a very highly reliable neutral change. And I'm looking at cumulative data. Cumulative data means that if the system would be totally right, my prediction method, and I start with a data set that has effect here in red, then all of that should show up as an effect. It should all be predicted somewhere here. So cumulative, I ask, what fraction in my data set has a score above a certain value, an effect above a certain value, okay? That's the cumulative fraction shown here. If I started with a data set that I know is neutral, and my system worked very well, then this should be my outcome, okay? Let's look at reality. This is the training set of the method. Effect, neutral. And really missing my pointer here. Somehow the difference, so ideal would be like this. The difference here is the mistake for the positives. The difference here is the mistake of the negatives. And in between, the degree to which it can pull red and green apart is the power of the method. Because this is a data set of neutral, and this is a data set of effect. If they would be on the same line, the method would have no distinction power, right? So that's the full distinction power it has. Now, what if I trigger that system with a data set of disease mutations? So now we're looking at mutations that we know that the carrier of that particular mutation has a disease. Irretrievably. You have that mutation, you have that disease. Okay? How do they look? And that's work from Chris Schaefer. Those are the disease mutations. For us, that, by the way, the last time somebody asked about machine learning, here's an interesting side effect. None in the orange data set had been used for training. None was similar to anything that had been used for training. Nevertheless, there's a signal stronger than the training set. How can that be true? Anybody who has ever used machine learning will know that you can never do better than training. How come? I have a data set that is better, for which the method is better than for the training set. Anybody use machine learning? Anybody, any idea?
Possible if you will use, if you will fit more than it's needed, then it can be, then in some cases
Overfit.
you will have more error.
But actually, wait. So when you overfit, then in some sense it's like learning by heart. When you learn by heart, you cannot do very well a new task. You cannot do something that you have never encountered before. Orange is a data set that you have never seen before. So if you learn by heart, you should still be better on red than on orange. Things you have not seen before, you should be less accurate at classifying than things you have learned by heart, right? So overtraining would show exactly the other direction. The more overtraining I have, the more the red would move up, but it would not be true because it wouldn't work for things it has not seen before. But that is not the case here. So again, how can that happen in machine learning? In fact, again, I have 20 years of machine learning experience. It had never happened to me before. It's the first time that happened. But any sense what could cause that? It's a consistency issue. So it means that the data set that you use for training of effect is not consistent enough, it's not clean enough. There were mistakes in here. And this data set obviously was cleaner. How come it's so clean? Well, diseases are a very, very clear effect. Is simply the intuitive answer here. So now that means that we have a method and we can answer what the difference is in the 20,000 of us. How that differs, whether they are meaningful. We expect that most of those 20,000 are somehow neutral. Most of the things that make us differ are not of the disease type. Because we all walked in here, right? The simplest definition of we are not sick is nothing has killed us so far. Nobody in the room is dead. I claim, right? And that's a clear definition of healthy. It may be funny, but you know, you're healthy. So where are we? Where's the variation between us? Where are they going to be on this spectrum between red and green? Anybody? You have no idea. Nobody knows. Okay. So they fall somehow exactly in between. And that, in many ways, is surprising because, in fact, it means that many of the mutations we have here, 25% of the mutations are predicted to have an effect above 40. And we believe that is a strong effect. I will later show you the line here. I believe it's 12% that have an effect that is almost as much as the disease mutations. So that's a real strong effect. That's a lot. Okay, how would it look if I compared us to Neanderthal? Or in this particular case, what I'm going to show you is Denisovan. So now I'm going to look for every single protein that we have, that Denisovan, this older than Neanderthal, has, and ask the differences between us. Do they matter? Okay? And that's what we see here, the early human. So that's more neutral. Let's get back further in time. Gorilla and we. Even more neutral. And here, by the way, again, not on machine learning. Now suddenly we have a data set that is below the neutral training set. Right? Again, because it's a very clean data set. We go further to the mouse, and that's, I believe, where we stop. That's more neutral. So So let's get one thing straight here. What I'm looking here at in the differences between us, we all have 20,000 proteins and essentially have the same 20,000 proteins, and we're looking at differences for every single one of our proteins. Here, I'm looking at a tiny set of proteins. Or tiny is not quite right, but this is a set of, in the ballpark of less than 10,000. So it's a much, much smaller data set. And a lot of changes in those. This is a small set with big changes. That's a large set with small changes. Okay? We're not, when I say more neutral, it's not meaning that the mouse is more similar to me than any of you in the room. That is not what I'm trying to say. But I'm saying, however, what that implies is that the mutations between us as a group, as a population of mutations, are more effect mutations than what is frozen in time. At some point in time, we had an ancestor with a mouse. What the speciation, the change from that ancestor to whatever led to mouse and whatever led to us, is a frozen change. That frozen change can no longer change, and that, in fact, is more neutral. While this here is a living population. Okay? And we clearly see that the mutations that we have in this living population, they do matter. In fact, Jana Bromberg had a compelling case in which she argues that the mildly effect mutants are the ones that define individuality. That's a PNAS paper that claims that. Now, let's get back to the issue of, in some sense, part of what you asked in your question, can we design a good gene or something like that? So, effects. I said a lot of those things between us will have effects. But effects can be bad and good. Let me give you an example of the sickle cell disease. Sickle cell anemia. And that, in fact, is one of those diseases where a single amino acid change in hemoglobin, is a particular protein, that is the E that is at position 6 in the changed version, in the disease version to V. This single change causes that disease. It's a really bad disease. You absolutely don't want to have it. Now, sickle cell anemia is much more prevalent in Africa, and in fact in malaria areas, than it is in Europe. And you see where that is going. That bad mutation, which causes a bad disease, sickle cell anemia, in fact gives you resistance against malaria. Okay, we have the same point, the same mutation that causes something that is really bad and really good. How can you say now bad or good? How can you classify? Well, one classification is you die, right? That's a simple one. But this is not that far away from it. Sickle cell anemia is a disease that shortens your lifespan, but malaria shortens your lifespan even more. So sometimes something that is bad can be better because there's something else that is worse that it prevents. And how can you see that in an organism? On the level that I talk about here, I predict the molecular impact. For me, there is no difference. I cannot see that this helps in malaria or that this causes a disease. All I can say, this is going to have an effect. This is going to change something. And the change could be for better or for worse. Now, what if we could somehow curtail, so those are the differences between us, and you see that's the disease mutations here. Let's look at the very bad ones here. Let's assume we could somehow classify them. Some of them are bad here, and some of them are good. And again, the question is, can we, through personalized medicine, possibly curtail the bad ones and the good ones? One drug tailored for each, and I already told you that doesn't work. But, and we get to the point that I said before, diagnosis and choice in food. Those things we can do. I can change the way I eat. I can change the way I live. I can choose my drug. So I believe the last time I said that there was one in the room who looked at me as if that was not a clear statement. So for instance, I have a high blood pressure. And there are particular drugs, medications that I take a particular drug every day. And so that regulates my blood pressure. And there is a particular drug for blood pressure, for hyperthermia, that in fact kills people with a particular mutation. Okay? So if you have that genotype and you take that drug, that is dangerous. For everybody else, this drug is fine. It's one of those drugs that came on the market and was fine for most people, but suddenly in the large group of people that were tested, they found, in fact, people with this mutation, and that became a killer, and the drug was retreated. And again, we are talking about $2 billion. So the moment you get to this point where you, in fact, are on the market, you lose a lot of money if you then have to retract that. And that, again, is something that you could have seen before, possibly. Certainly, we can keep it on the market as long as we know that there is some subset of us who shouldn't take it, right? Diagnosis is maybe ultimately the way cancer types, or again also drugs, are classified today is because you have a certain age, because you have a certain gender. You're a male, I'm a male of 55 years or something like that, and that is all that is ever taken into account. That's today's most sensitive way of looking at people. Or I'm getting a certain drug because my physician happens to have the best experience with those drugs. This gives us an access to much better choice, much wider way of looking at it, and that could help us to in fact curtail. So it's not that we change the molecular effect and turn one into good or bad. It's just the way we live with it. And that is part of diagnosis. And that, in fact, is something that will happen and that can happen. So our genetic differences clearly matter. And many of them are really bad. They make our individuality and they make us sensitive to problems. So many of these changes that we have, the 20,000, in fact will be bad for me. But evolution doesn't care. Evolution cares for a species. If I have a mutation that will kill me early, as long as I pass on my genomic material, that what is important is that anything that could be really bad for me, if the environment changed, may be good for my offspring. So something that is bad for me, if it has the chance of bringing some advantage to the offspring, is something that evolution needs. So as a species, what I'm saying is we throw mutations into the living pool of us that are really bad for every single person in the room. But who cares? We in the room, I mean, you care, I care for myself. But evolution doesn't, because if this change, like in the sickle cell anemia, the malaria, if that single change has an advantage, then that may make the difference for the next species, or for not the next species, for the next generation. So we survive as a species, not as an individual. So somehow the great news here is that by curtailing that end here, through personalized health, in some sense, we can combine the good and the bad, and we can in fact harness the power of diversity. We can combine the best of both worlds. So we can use our difference and live differently, and have the advantage for ourselves from the difference and for the species. That's a great future, right? And in fact, I believe really that is something that we are about to witness. And by the way, there's a TEDx talk that gives part of that story from me. It is going to be challenging, and it's a long way to go to beam from individual SNPs to health. And it needs the connection of many resources, and there's a lot of work to do, and we cannot do it alone. But some steps are being done already, and I'm very certain that over the next few years, the genome, when you walk to a physician, your genome will be there, and some actions will be decided. And most of those will be for the better. Okay, so where are we standing here? So today we talked about sort of an overview over the entire lecture in some sense. And on Thursday I'm going to talk about protein function really in more detail. And next week Tobias Hamp is going to introduce a particular method that predicts function. In principle, so the week that I was sick is a little bit in the way of the schedule. I cannot do it next week. So this should have actually really been in 2 weeks from now, but I hope you will be living with that. Again, for everybody, the lectures will all be in this room, Thursday and Tuesday. Thanks for your attention.